So the structure of double-strand DNA is double helix. And this classic double helix, you'll see, here's our backbone, right? And then in these planar flat rungs on the ladder, those are our base pairs. But what you'll notice is this is not a perfectly symmetrical double helix. There's what we call a major groove, this larger space here. If you had this, if it was sort of like a Twizzler, you could feel this major groove and it would be wider than the minor groove. So it's not perfectly symmetrical. Here's our, these are the sizes of these things. But what we call B-form DNA has a major and a minor groove, and this shape and this size. Just by the way, the major groove is often a binding site for things like proteins that bind to DNA, the larger one, whereas the minor groove, the smaller one, is often a small molecule drug binding target. That's one of the consequences of this. So the B-form DNA, again, with a major and a minor groove, and here the backbone is shown in purple, the base pairs are shown in red. This is the most physiologically relevant form of DNA. This is usually what we think of as DNA when we're thinking of the shape of it. The helices, it turns out, can be classified as right-handed or left-handed. B-form DNA is a right-handed helix, major and minor grooves. And please notice the backbone is smooth. It's not zigzagging around or anything, smooth. And the base pairs are planar. They go straight across here, okay? But it turns out there's other forms of double-strand DNA. There's an A-form DNA, and when you watch the movie, please pay attention for A-form and B-form DNA. The A-form is more compact, still a right-handed helix. It's got less water in it, and I'll show it to you in just a second. There's also something called Z-form DNA, and in this case, the Z does stand for zigzag. The backbone is not smooth in this case. It's a left-handed helix, and this was, it was proposed to exist before it was actually found. Rosalind Franklin did not know about Z-form DNA, but it was found to be able to form when you have, in particular, alternating purine-pyrimidine sequence such as GC, GC, GC. Okay, so here's the three forms of DNA that we think about the most when we talk about DNA structure. B form in the middle, right-hand helix, smooth backbone, planar or straight across, what am I trying to say, base pairs, shown here as well. Smooth backbone, straight across, planar base pairs. Here's our A-form DNA, okay, and this is the same length of DNA, so you see it's a little more compact. We still have, moving over to here, we still have a smooth backbone, but notice that we don't have perfectly straight across base pairs in this case, a little bit skewed here. Here's Z-form DNA, and in this case, you'll notice here's B-form in the middle, right-hand helix. Here's Z-form on the left, left-hand helix, and it's not smooth, zigzag, zigzag, zigzag. And so these, again, just introducing you to this, these are the three forms that we talk the most about when we talk about the structure of DNA. We will talk mostly about B form, and really this is us talking about it right now. But if you have double-strand RNA, just by the way, it tends to form, in general, something that looks more like the A form, okay? Okay, now, DNA can be modified. This is an extraordinarily complicated topic to discuss at this moment in your scientific career. I am going to not just simplify this, but completely oversimplify this. But as you know, DNA is chemical, right? The bases can be methylated, excuse me. Sorry about that. DNA can be methylated. One of the common sites in mammals are called CPG sites or CPG islands or CG sequences. So when you're looking at DNA, which is really long, if you come across a C followed by a G, sometimes in mammals, the C, the cytosine base, is modified with a methyl group to be 5' methyl cytosine. Please don't worry about the chemistry of this, but here's the point. It's methylated. And so sometimes we see these things we call CPG islands, which are unmethylated. Now, I'm truly oversimplifying this, but sometimes the methylated regions of DNA are transcriptionally silent. The genes are turned off. Unmethylated regions, and again, I'm generalizing, and this is not always true, unmethylated regions can be transcriptionally active, or the genes are turned on. So this is one of the ways to turn on or turn off regions of DNA. Now, it's more complicated than just CPGs. A's can also be methylated, and DNA methylation is a type of what we call an epigenetic mark. Okay, your sequence is your sequence, but you can also have epigenetic outside your genetics where regions can be turned off or turned on as a result of environment or other signals. There's enzymes that methylate DNA. In general, the general name for it is a DNA methyltransferase. There's at least 3 of these in humans. And methylated DNA, in general, less transcriptionally active. But I'm seriously simplifying this topic. In addition, and that was not the only thing, not the only modification that can happen to DNA. In addition, DNA is compacted. So DNA molecules are really, really, really long, and we have to get them to fit into the nucleus of a cell. You may have heard that the DNA in one of your cells is actually something like 3 meters long, but of course it's very, very thin, but it has to be compacted. The analogy for getting DNA into the nucleus of the cell is it's analogous to squeezing 60 miles of wire into a basketball. Okay, so we never find DNA naked in a cell, but we always find DNA complexed with proteins. And the major DNA proteins in eukaryotes are called histone proteins. Histones do a whole bunch of things, but one of their roles is to help compact the DNA, helping the molecule get shorter, not quite literally shorter, but by wrapping around things. And so you'll hear the term chromatin, right? Not chromosome, but chromatin. Chromatin is how we find DNA in the nucleus, and it's DNA complexed with histone proteins. So if we were going to isolate DNA, if we isolate not only the nucleic acid, but also the protein, we would be isolating DNA plus histones and some other proteins, okay? When we talk about isolating DNA, you'll see that in the methods we're going to do, we often remove the proteins, but you don't have to. There's other methods. Histones, besides serving to compact the DNA, also serve to regulate the chromatin, or in other words, turn the genes on and off. The histone proteins themselves can be modified. One example, and again, really complicated topic that I'm completely oversimplifying, but in the protein histone H3, there's a lysine residue at position 9. If that is methylated, that's associated in general with transcriptionally silent chromatin. In other words, if the histones in a region have lysine 9 at methylated in their H3, in their histone H3, then the gene may be turned off. But there's a number of types of histone modifications, and what can happen is what we call chromatin remodeling, changing the modification. So this is a really, really complicated topic that I'm simply letting you know it's a thing. So simplifying this even more, DNA is complexed with histones and other proteins. The DNA molecule itself can be modified by, the only thing we mentioned is adding a methyl group. That's not the only thing that can happen. That can regulate whether the gene in that region is on or off. The histones are also modified. They can have a number of different modifications as well, and that's another way to determine whether a region of DNA is transcriptionally active or turned on or turned off. The DNA is compacted into nucleosomes, packaged into chromatin, and eventually also into chromosomes, or the visible chromosomes. And the DNA and the histones are chemically modified. It impacts whether the genes are on or off. Crazy complicated story that I'm simply letting you know you'll probably learn more about in the future. So here's our sort of what a nucleosome looks like in a schematic on the right. In humans, there's about 146 nucleotides of double-strand DNA wrapped around a histone octamer. Now we're talking about a protein. The octamer has 8 subunits. H1 is not in that core histone, not part of the octamer, but the octamer has 2 molecules of H2A, 2 molecules of the protein H2B, 2 H3s, 2 H4s. That's typically shown here, 4 on the top, 4 on the bottom. So the blue is the core, H2A, H2B, H3, H4. The green is H1 protein. And then here is our double-strand DNA wrapped twice approximately around a nucleosome. That's what we call the core nucleosome. We're going to have, so here's our DNA helix, and we're showing it naked. You can now look back at the slides, and there was no mention of proteins. But here we have these core nucleosomes, these sort of flat discs. And here's our DNA wrapping around it. And so this is going to compact our DNA. Now we form, sometimes they call these solenoids, but now we have a number of these nucleosomes compacting together. Notice that we're getting wider, even though the artist is not showing it to scale because they can't, because it's too big. Now we take these, form them into looped domains, further condense them into what we call condensed coils, and eventually we get to what you could see in a light microscope, one of these big fat metaphase chromosomes. This is, you'll see this in textbooks, but obviously it's simplified. It's not so straightforward. Here's another artist's rendition of this. Here's our double helix. Here's our histones. Here's our core nucleosomes. Here they are, compacting more. I like to say, and then a miracle occurs and we have a chromosome, because it's not very obvious how this happens here. And this is still a process that's an active area of research. Yet another one, skipping the naked DNA, but here's nucleosomes, coiling, magic happens, whoops, sorry, and we have a chromosome, okay? Let me pause here, and I think we'll only have one more video.